In Lab 6, we're going to be taking up the gram stain and the capsule stain. Again, we have a number of videos to watch, pre-lab quiz videos, along with the soft chalk lesson on gram stain results. And of course, you have your Lab 6 pre-lab quiz to do that must be completed at least 2 hours before the start of your lab. And also remember, of course, when you're finished with the lab, to answer the performance objectives at the end and take the self-quiz. So we'll start out with the gram stain. Now, the gram stain is the most widely used stain in bacteriology, and it's what's called a differential stain because it differentiates gram-positive bacteria from gram-negative bacteria. And of course, as we learn, gram-positive bacteria stain purple with the gram stain, while as gram-negative bacteria stain pink. And keep in mind, gram-positive and gram-negative have nothing to do with charge. It simply designates two different groups of bacteria. Now, the reason gram-positive and gram-negative stain differently is because they have fundamentally different structures in their cell walls, as we learned in Unit 1. So remember, the gram-positive cell wall, when we look at it, has a thick, rigid layer of peptidoglycan, which is actually many layers of peptidoglycan all sandwiched together, which surrounds the cytoplasmic membrane, which of course encloses the cytoplasm, and the space between the membrane and the cell wall is called the periplasm. And in our illustration of that, we can see here again that the gram-positive cell wall has many layers of peptidoglycan sandwiched together, and interwoven amongst the peptidoglycan are lipoteichoic acids and teichoic acids. Now, on the other hand, the gram-negative cell wall looks multi-layered because it has a thin layer of peptidoglycan that we see here surrounding the cytoplasmic membrane, but then it has another membrane on the outside called the outer membrane. And of course, the space in between the membrane and the wall is a periplasm. And if we look at the illustration of that, we see again that typically a gram-negative has only a couple of layers of peptidoglycan surrounding the cytoplasmic membrane, a thin layer, but it has a whole nother outer membrane that surrounds the peptidoglycan, and that's composed of phospholipids on the inner layer, along with lipoproteins that connect the outer membrane to the peptidoglycan. And then the outer layer of the outer membrane is primarily lipopolysaccharide, or LPS. And of course, all bacteria have surface proteins. And don't forget, the gram-negative cell wall has pore-forming proteins called porins that form pores that let materials cross the outer membrane in order to enter the bacteria. So it's these differences, as we'll see shortly, that allow gram-positive and gram-negative bacteria to stain differently. Now, one of the first things we have to realize about the gram stain is that it's not a stain, but a 4-step staining procedure. So first, we stain the bacteria with the basic dye crystal violet. And a basic dye, remember, gives you a direct stain. So after step 1, both gram-positive and gram-negative bacteria are simply directly stained and appear purple, the color of the crystal violet. The second step is we treat the bacteria with Gram's iodine solution. And this allows the stain to be retained better in the bacteria because the crystal violet combines with the iodine to form an insoluble crystal violet iodine complex. And that tends to be held within the cell better. After step 2, both gram-positive and gram-negative bacteria still remain purple. So the differential step is step 3, where we add Gram's decolorizer. And we're just going to use ethyl alcohol. Sometimes it's a mixture of ethyl alcohol and acetone. Now, what happens here is that the gram-positive bacteria retain the crystal violet iodine complex and remain purple, but the gram-negative bacteria become decolorized or unstained. They lose the crystal violet iodine complex. So after step 3, gram-positives are purple, gram-negatives are now unstained. So finally, we add the counterstain safranin. It's a basic dye like crystal violet, but it's pink rather than purple. Since the gram positives are already stained purple, they remain purple. They're not affected by the pink counterstain. But the gram negatives, which are colorless after step 3, pick up the crystal violet, or pick up the safranin and stain pink. So when we're all done, the gram-positive bacteria, like the Staphylococcus epidermidis we're using today, is gram-positive, whereas gram-negatives, like the E. coli we're staining today, stains pink. And there's a Flash animation I'm going to show you. Flash doesn't play well on modern browsers anymore, but I've set mine so I can show you this one animation because there's not another HTML5 version of this, since it's not one of mine. But this shows you step by step how the gram stain works. So if we start out, of course, the idea is that gram positive will stain purple, gram negative is pink. Let's start out by looking what happens with a gram positive bacteria. Now, as we see, that has a thick layer of peptidoglycan, many layers of peptidoglycan sandwiched together, interwoven with teichoic acids and lipoteichoic acids surrounding the cytoplasmic membrane. So when we add the crystal violet enters the bacterium, and the bacteria becomes stained purple. Remember, this is a basic dye, so the positive ion containing the colored crystal violet combines with the negative charge in the bacterium. So that's simply a direct stain, and the gram-positive look purple after this. Then we add the grams iodine. Now the iodine, when it enters, combines with the crystal violet, as we see here, to form a larger insoluble crystal violet iodine complex, which we see happening here. And so being a larger insoluble molecule, it tends to be retained by the bacteria more readily, stay inside. And again, the gram-positive look purple after this step. When we add the decolorizer, in this case alcohol we're going to use, one of the characteristics of alcohol is that it's a dehydrating agent. It removes water, and in this case it's removing water from between the layers of peptidoglycan, causing the peptidoglycan to condense, and that condensation of the cell wall helps to hold the dye in better. So the crystal violet iodine can't really get out of the dehydrated multi-layered gram-positive cell wall, and it remains purple. It retains the crystal violet iodine complex. When we finally add safranin in the final step, although the safranin enters the bacterium, the pink is much more obvious than the purple. The pink doesn't really appear, and the gram-positive still look purple when we're done. So when we're all done and look at it under the microscope, the gram-positive bacteria will appear purple. Now, if we compare what happens with a gram-negative bacterium, remember this has a very thin layer of peptidoglycan, only a couple layers thick, but it has an outer membrane of phospholipid, lipoprotein, and lipopolysaccharide, as well as porins. When we add the crystal violet, like in the gram-positive, the crystal violet enters the bacterium, and the positively charged crystal violet iodines interact with the negatively charged components like proteins and DNA in the bacteria, and the bacteria look purple after step 1. Again, simply a direct stain. When we add grams iodine, like in the previous case, the iodine enters a cell, and it combines with the crystal violet, forming an insoluble, larger crystal violet iodine complex that's better retained by the bacteria. But here's where we see the big difference when we add the decolorizer, the alcohol that we're using. Now, another characteristic of alcohol is that it's a lipid solvent. And remember that outer membrane is composed of phospholipids, lipoproteins, lipopolysaccharide. So the alcohol, when we add it, actually dissolves away the outer membrane. And although there may be some minor dehydration of the peptidoglycan, it's only a couple layers thick rather than many layers thick. So it's really not enough peptidoglycan to retain the crystal violet iodine. So with the outer membrane gone, the crystal violet iodine complex leaks out of the cell, and the gram-negative becomes unstained after we add the decolorizer. So finally, when we add the safranin in the final step, since the gram-negative is now unstained, it will pick up the pink safranin. And when we look at it under the microscope, the gram-negative bacteria will stain pink. So that's the mechanism behind the gram stain. And here we have it stated: the alcohol acetone causes dehydration of the multilayered peptidoglycan, decreasing the space between the molecules, causing the cell wall to trap the insoluble crystal violet iodine complex within the cell. But in gram-negative bacteria, the alcohol or alcohol acetone mixture, being a lipid solvent, dissolves the outer membrane, may also damage the cytoplasmic membrane, and so as a result, the crystal violet iodine complex is leached out and it becomes decolorized. Now, it's also important to point out that gram positivity in a gram stain is simply the ability to retain the crystal violet iodine complex. And that's not an all or nothing phenomenon, but a matter of degree. So there are several factors that can give us faulty gram stain results. And we have to keep these in mind today to get reliable gram stains. And since we are doing gram stains in a number of labs coming up, we need to get to the point where we can do reliable gram stains. So every time you do a gram stain, you want to kind of pay close attention to what you're doing each step of the way, and if it doesn't work out right, know how to modify that next time, so eventually you learn how to do good gram stains, since there's a little bit of an art to that. So one thing that can go wrong would be, of course, the methods and techniques used. If we overheat during heat fixation, that can damage the cell wall so that even gram positives lose the crystal violet. You can over-decolorize with the alcohol. That's why the timing and watching it carefully is very important, because you can over-decolorize. And that's also where we're going to see the number of bacteria we add can be important. If there's too few bacteria on the slide when you make your smear, it'll be very hard to find them when you focus, and it'll be much easier to over-decolorize. If there's too many bacteria on the slide, then of course you won't be able to see individual bacteria, and you might under-decolorize. And you even have to be careful that you don't do too much washing between steps, because all of that can sometimes cause some gram positives to lose the crystal violet iodine complex. Another thing we have to watch is the age of the culture. As the cells use up all the nutrients and waste products accumulate, that can start to damage the cell wall. So cultures more than 24 hours old may lose their ability to retain the crystal violet iodine complex if they're gram positive. So we're always providing you with fresh cultures that are less than 24 hours old. And even the organism itself, there are some gram positives that retain the crystal violet iodine complex better than other gram positives, but the ones we're using today should work fairly well. But we do have to use very precise techniques and pay attention to what we're doing. So the organisms for the gram stain, you're going to be using plate cultures of Escherichia coli. We've seen this a number of times now. That's a small gram-negative bacillus. And we're also going to do a gram stain of Staphylococcus epidermidis. We've also looked at that a few times. That's a gram-positive coccus with, of course, a Staphylococcus arrangement. And again, I've listed the videos here on how to heat fix, how to prepare a slide for staining, how to do the gram stain, etc. These are the ones you had for your pre-lab quiz, but you might want to review those again. So what we're going to do first is a gram stain of E. coli, and first we have to heat fix a smear of Escherichia coli. And again, it tells you step by step how to do that, as we showed you in lab 5 when we did the direct stain, including the actual gram staining. But again, for preparing the smear, make sure you definitely go back and read how to prepare or watch the video on how to prepare a slide for staining when taking bacteria from an agar culture, and how to heat fix the slide. Because again, there's a lot of little tips along the way. Remember, the amount of bacteria is very critical. We want to make sure also we label the slide so we know which bacterium is on which slide and which side of the slide is up, so we don't accidentally turn it over during the process. Remember, you want a very small drop of water. The more water you add, the longer it's going to take to air dry. Remember, we want to add just enough bacteria by touching the loop to the drop of water until it becomes definitely visibly cloudy in the water. So we want to make sure there's enough bacteria there that we're going to make it pretty easy to find under the microscope, but not so much that the whole drop appears cloudy like this, because then they'll be piled on top of one another. And again, if you have them piled on top of one another, you're not going to be able to decolorize properly. But if you have too few, because there's so few bacteria losing dye, you might actually over decolorize. So the amount of bacteria is really critical in a good gram stain. And then, of course, once it becomes just visibly cloudy, then you're going to incinerate the rest of the bacteria from the inoculating loop, let it cool, spread the bacteria that are in the water over the slide to get a thin film. And again, that should cover most of the slide if you have the right amount of water. And then you want to let that completely air dry. It has to be air dried before you heat fix it, remember? So when it is air dried, that should look like a thin powdery film. You should be able to see it with the naked eye, or you probably don't have enough bacteria, but you should be able to see through the smear, indicating that you don't have too many bacteria. And then, of course, we're going to heat fix it by holding it bacteria side facing you towards the over the opening of the Bacto incinerator for 10 seconds. At that point, you're going to stain the bacteria with Hucker's Crystal Violet for 1 minute. That's the purple dye. So you want to cover the whole smear. After 1 minute, pick the slide up and gently wash with water. So remember, we're going to direct the water above the smear, let it run down over the smear until the liquid at the bottom turns relatively clear. We're just washing the excess dye off the glass, not off the smear. So don't look for the smear to lose color, just look for the dye coming off the bottom to become clear. Now, you don't have to blot dry between steps. You're just going to shake off the excess water, but don't blot it. Immediately put the slide back on the staining tray, flood the smear with grams iodine, and leave that on with 1 minute. Then pick up the slide and gently wash with water. Again, don't blot dry. And then we're going to decolorize by picking up the slide and letting the grams decolorizer run down the slide until the purple just stops flowing from the bottom. And that's really the critical step you have to watch. So as it showed you in the video, we're going to let the alcohol run over the slide. And as the video shows you, we're going to take the edge of the dropper bottle. You don't really have to squeeze the big bottles we have. Gravity will pull it out, but just keep going back and forth over the top and let it run down over the smear. But remember, you're watching the bottom of the slide, not the smear, the bottom of the slide. Very carefully. Keep going back and forth, watching the bottom carefully. And as soon as the liquid coming off the bottom looks clear or maybe even a very light blue, immediately stop decolorizing and immediately wash with water. So make sure your water bottle is nearby so that you can immediately wash it with water. And then finally, once you wash it, again, just put it back on the staining tray, flood the smear with safranin, the counter stain, leave that on 1 minute. That's our pink counter stain. After 1 minute, pick up the slide, wash that gently with water, and then put it in your book of bibulous paper. Move it around several times to gently blot it dry. Remember, just lightly rub your fingers over the book of blotting paper with the slide inside. Don't push too hard because you don't want to break the slide. And if you've done it right, you should actually be able to see with the naked eye at this point, just a very light colored staining film where you can see there's definitely bacteria on the slide. Now let's review that critical decolorizing step once more from the video that we have, because that again is the make it or break it. And this will also be demonstrated in lab prior to you doing the gram stain. Wait for it to load in here. So notice the slide's labeled. This is E. coli. We've already stained it with crystal violet, and we've treated it with Graham's iodine. And notice you can just barely make out a slight color on the slide that shows you there's definitely bacteria there, but not so many bacteria that you can't see through it. So that's, again, a good amount of bacteria to have. So note we're going to be touching the edge of the dropper to the slide, going back and forth. We want to go side to side so that we make sure the whole smear gets decolorized. We don't want to miss decolorizing any part of that smear. And again, you don't really have to squeeze the bottle. Gravity will let it pull out pretty good. So let's watch the decolorizer come down over the smear. And remember, you're going to watch the bottom of the slide here, not the smear, but the bottom of the slide. There's the purple. So that's the procedure for the Gram stain. Your first Gram stain will be E. coli. Then you're going to make a second Gram stain using the same exact procedure of Staphylococcus epidermidis. Now you want to get both slides going at the same time. Prepare the smear of E. coli, get that heat fixed, prepare the smear of Staph epidermidis, get that heat fixed. That way, if you're doing them both at the same time, they can be air drying at the same time, which is going to save you time rather than doing one slide all the way through and then starting all over again and doing the second slide. So get the heat fixed smears made at the same time, but then make sure you stain them one at a time. Don't try to stain them both on your staining tray at the same time because you won't be able to time it properly or watch the decolorization properly. And make sure you only have one slide on the staining tray when you're staining. Don't leave the other slide on there while you're staining another one, because if you bump the tray, you may knock the other slide into all the wastewater and dye and ruin the slide. And once you're done staining it, put it on the lab bench. Again, don't leave it on the tray because you could bump the tray and knock your finished slide into all the dye and wastewater. Once the two slides are finished, then you can clean up and give yourself some work area. And make sure that you pour the used dye down the sinks at the 3 lab benches that don't connect to the sewer system. That's all collected, remember, in a carboy, so we don't cause any pollution with the waste dye. So make sure you don't put the dye from your staining tray, the wastewater down the sink, but rather put it down one of the designated drains in the 3 lab benches. And once you've put stuff away, then you'll have a nice work area. And after reviewing your focusing steps for oil immersion, then you can look at your 2 gram stains. And again, you'll be showing me each of these gram stains. Make sure that when you show it to me that you have a good area with isolated bacteria where you can tell the shape and arrangement. And I will be asking you if it's gram negative or gram positive, and be asking you the shape or the arrangement. So those are the 2 gram stains that you'll be doing. Now, depending on the instructor, you may or may not do the capsule stain. In my lab, I'm going to set out some demonstrations of the capsule stain, since we don't need to do another capsule stain during the semester, but we do want to do a number of gram stains. So we want to concentrate on you doing 2 gram stains apiece. But let's talk about the capsule stain and capsules. Of course, we learned about capsules of glycocalyces back in Unit 1. And as we saw, that's a slimy, viscous covering that surrounds the bacterium. And when it's rather extensive and adheres to the bacteria, we call it a capsule. So this is the capsule stain we'll be looking at today of Klebsiella aerogenes, which used to be called Enterobacter aerogenes. And in the capsule stain, we're staining the background. We're letting the dye dry on the slide, and we stain the bacteria, but the capsule doesn't pick up the stain. So we can see the clear white capsule surrounding the purple bacillus. And by having the background in the bacterium stain, we can see the capsule nicely. And of course, on a practical, if I ask you what structure do we see on this slide, that structure would be the capsule. Remember, we can stain bacteria in order to observe structures such as capsules, endospores, flagella, and such. In this case, we're looking for the structure that we call the capsule. And as we learned in lecture, capsules allow bacteria to resist phagocytosis by the host phagocytic cells. They also allow bacteria to adhere to surfaces to resist flushing. And of course, we also learned that many bacteria produce biofilms, a population of bacteria that are adhering to the host cell and embedded in a common capsular mass. Now, if you do the capsule stain, you're going to be making a capsule stain of Klebsiella aerogenes. We're growing it in skim milk broth. The skim milk provides nutrients for capsule production, but when the milk dries on the slide, the proteins in the milk pick up the stain to provide a slightly stainable background. So if you do this, this will be growing in a skim milk broth culture. So the first thing you want to do with your loop is stir up the culture so we get the bacteria distributed, or else vortex it on the vortex mixture for a few seconds. And then place 2 to 3 loops of Klebsiella aerogenes on a clean microscope slide, of course using aseptic technique. Then spread the sample out to cover a good portion of the slide. You should be able to cover about 1 inch of the slide with 2 or 3 loops. Make sure you let it completely air dry. But we're not going to heat fix the capsule stain. The capsules stick quite well to glass, and the heat could damage the capsule. So this one we're just going to let air dry but not heat fix. You're simply then going to stain it with crystal violet for 1 minute, but we're not going to wash off the crystal violet with water. Rather, we're going to wash off the excess crystal violet with 20% copper sulfate solution. So you're going to let that run down over the smear until the purple stops running off. Then shake off the excess copper sulfate solution, put it in your book of blotting paper and blot dry, and observe using oil immersion microscopy. And both the organism and the dried milk on the slide pick up the purple crystal violet, whereas the capsule will remain colorless, as we showed you in the illustration here, where we see the clear colorless capsule surrounding the purpley stained bacillus. And here's a photograph of another capsule stain, this time of Streptococcus lactis, one of the normal microbiota in milk, where you see the chain of streptococci with the clear capsule around them. So again, on any slide where you see a capsule, and I ask you what structure do we observe on this slide, you'd respond with capsules. So recognize capsules under the microscope. Then you'll record your results here for the gram stain of E. coli, indicate the color it stains, whether it's gram positive or gram negative, and the shape. For the Staphylococcus epidermidis, again, indicate the color that it has stained, whether it's gram positive or gram negative, the shape, and the arrangement. So again, remember, Escherichia coli is a gram negative bacillus, and Staphylococcus epidermidis is a gram positive coccus with a Staphylococcus arrangement. And then on the capsule stain, you can record your results here. Here's the capsule stain of Klebsiella aerogenes that we showed you, and the one of Streptococcus lactis that we showed you. So if those are done as demos, make sure you record your demo slide there. And of course, at the end, as always, is your performance objectives and your self-quiz.